Today we're going to talk a little bit about cellular neuroscience. Neuroscience is how we interact with our environment and with each other. Everything that you've ever learned about psychology, it happens because of our nervous system. The types of neurons in our body can be broadly defined as sensory, interneuron, and motor neurons. Sensory neurons receive information from the periphery, from the environment, and interneurons are housed entirely within the central nervous system of our brain and spinal cord. Motor neurons, also called motor efferents, send information from our central nervous system to the periphery to allow behaviors to occur. There's no such thing as a typical neuron, and yet we are going to pretend this whole semester that there is a theoretical pretend typical neuron. This is what our pretend neuron looks like. It is a multipolar neuron, meaning that it has a bunch of dendrites coming out of the cell body or soma. These dendrites have what appear to be little bumps on them. These are called dendritic spines. They are specialized structures that are designed for synaptic transmission, to receive synaptic information from upstream cells. The cell body connects to this long thing here called the axon. Where the cell body connects to the axon, there's another structure called the axon hillock. And we'll talk more about the axon hillock when we talk about the action potential. The axon can be very long, all the way from your spinal cord to your tippy toe. Myelin is made up of another cell called a glial cell, and it wraps itself around the axon so that the action potential can travel faster while also saving the cell energy. And we'll talk more about that in another lecture. Messages in the neuron are received at the level of the dendrites and soma. And then at the axon hillock, the decision is made whether or not to send a message down to the terminal. If a message is sent, when it reaches the terminal, it can stimulate the release of neurotransmitter onto downstream neurons. This is an electron micrograph showing the location of the axon hillock. This area doesn't have any ribosomes, and it doesn't have most other cell organelles. It just has cytoskeletal elements and organelles that are being transported down the axon. Neurons can receive hundreds of thousands of upstream connections and can send hundreds of thousands of downstream connections to other neurons. Where neurons contact each other is called a synapse. The presynaptic cell is the one sending the message, and the postsynaptic cell is the one receiving the message. If we zoom in on a synapse, in this case we're looking at an axosomatic synapse, where the axon terminal of a presynaptic neuron is sending a message which is received at the soma of the postsynaptic neuron. When we zoom in here, we see that there are lots of specialized structures in the presynaptic neuron. Microtubules that have been used for exoplasmic transport of materials down to the terminal. Mitochondria to produce ATP locally. We have all of these circle things here, which are vesicles that are filled with neurotransmitter. At the release zone, we have neurotransmitters that are docked and ready to go. We have other vesicles that are docked and ready to go. We have other vesicles that are getting ready to go to the release zone, so that when we want to send a signal, we can send many neurotransmitters into the synapse. Neurotransmitters enter the synapse when vesicles fuse with the presynaptic terminal membrane. They will ultimately bind to receptors located on the postsynaptic membrane. This postsynaptic density got its name because if you look at an electron micrograph figure of a synapse, you see a very dense area on the postsynaptic membrane because there are so many proteins present for receiving the neurotransmitter and sending a message within the postsynaptic neuron locally. We can categorize synapses as axodendritic, axosomatic, and axoaxonic. Axodendritic synapses can either be on a smooth dendrite or on the dendritic spine, which again is this specialized structure made just for synapses. An axosomatic synapse is just like it sounds like. It's the terminal button of a presynaptic neuron making contact not with the dendrite, but with the soma of the postsynaptic neuron. Axoaxonic synapses are a little bit different. Here we have a presynaptic neuron making synaptic contact with a terminal button of a postsynaptic neuron. The axoaxonic synapse allows this blue neuron to tell this red neuron how much it should talk to this orangish neuron. While the cell body has to do all of the cell things for a neuron, many things have to make it down to the terminal. In order for things and stuff to get down to the terminal, we rely on axoplasmic transport. Stuff, like vesicles, has to go through anterograde transport to the terminal buttons, and then other stuff, like waste material, has to go through retrograde transport back up to the soma. This is another figure of the same information, but this figure came from Neuroscience Online, and it's showing anterograde transport walking some vesicles down to the terminal, and retrograde transport, some vesicles returning to the soma. The proteins that produce anterograde transport are called kinesin, and the proteins that produce retrograde transport are called dynein. Dynein is a slower process than kinesin. So things get to the terminal faster from the soma, and it takes longer to go the other direction. As we mentioned, the soma is the cell body, and it has to do all of the same things that any normal cell has to do. We need to have a nucleus with our genetic material so that we can do transcription and translation of RNA into proteins. We need to make energy in the form of ATP through our mitochondria, and all of the other things that any normal cell has to do, plus provide support for receiving and sending electrical information. This is just a list of intracellular organelles and some other useful vocabulary terms that you should be familiar with. If it's been a while since you've seen the inside of a bio textbook, it might be a good idea to go through these terms and just make sure you have a handle on some of just the basics of what a cell is. Because neurons, of course, are cells, but they have lots of other fun features too that most cells don't have. Most people have memorized that the mitochondria is the powerhouse of the cell. And that's not wrong, but it's important to know what that actually means. Mitochondria receives fuel sources in the form of glucose, fatty acids, and amino acids, and through a process, it produces this molecule called ATP, adenosine triphosphate. ATP is the energy currency of the cell. Protein synthesis begins with DNA in the nucleus. This DNA has to be read and converted into RNA. That process is called transcription. The RNA can then be translated into protein. Besides the neurons themselves, glial cells are an important part of our nervous system. For the most part, they don't form synapses. For the most part, they have only one type of process. That is, they don't have dendrites and axons. They just have one type of thing sticking out of them. For the most part, they're more likely to divide and compared to neurons. And for the most part, they are less electrically excitable compared to neurons. We have some different glial cells in the central nervous system compared to the peripheral nervous system. The main difference that I want to draw your attention to is that in the central nervous system, oligodendrocytes are responsible for making myelin. In the peripheral nervous system, Schwann cells are responsible for making myelin. One of the most diverse types of glial cells is the astrocyte. Astrocytes can contribute to this blood-brain barrier by wrapping around blood vessels so that they are not leaky. That helps to protect our brain from stuff that shouldn't be received in the brain. And we'll talk more when we get to chapter 4 of Carlson about the blood-brain barrier and how drugs can get past the blood-brain barrier even though we have our astrocytes helping to form it. As I mentioned, oligodendrocytes are myelin-producing cells in the central nervous system, and Schwann cells are myelin-producing cells in the peripheral nervous system. The function of myelin is the same whether it came from a Schwann cell or an oligodendrocyte, but the difference is structural. Here we have a single Schwann cell wrapping multiple times around an axon so that one Schwann cell forms one segment of myelin. In contrast, a single oligodendrocyte can form myelin segments on many neighboring neurons, and even multiple segments on the same neuron.